Hello students, we're going to apply what we learned from the previous video lecture regarding lipids to biological membranes. So this plays a really important role in biochemistry because it links to how molecules, important biological molecules, get in and out of the cell. So we first need to address what is the cell membrane, also known as plasma membrane. And then the second part of the chapter, not in this video lecture, but the next one, is how do molecules and ions move in and out of the cell membrane. So eukaryotic cells have membrane closed organelles in addition to the cell membrane, and it has membrane enclosed organelles such as the nucleus and mitochondria. For us, the mitochondria is a really important discussion later because it's going to play a role in how we process our energy molecules for energy, so like glucose and fats. So we'll come back to that idea. The molecular basis for the membrane structure is that it's primarily lipids and proteins. And the lipids form a lipid bilayer where the polar head group is in contact with the aqueous environment, either on the outside or inside, and the nonpolar tails are buried within the interior of the lipid bilayer. So what is driving the formation of these bilayers will be non-covalent interactions. And we'll talk more about this in just a second. So let's take a look at one picture, and then there's another one coming up here. This kind of gives us a little bit of a feel for what we're talking about. So the cell membrane has in it the lipid bilayer, which is shown here in the yellow, the bright yellow part. And then there's proteins that span the entire cell membrane. There's proteins that are face only on one side. There's going to be carbohydrates involved. So we'll come back to each of those in just a few minutes. But the overall structure is dynamic. So there's motion. Proteins are floating in the sea of lipids. They will have attractions for each other that keep them somewhat in place, but the word dynamic here means that there's motion. And then this whole thing is called the fluid mosaic model by Dr. Singer, who's at UCSD. And what is the role of this is to confer what's called selective permeability, that only certain molecules are allowed to flow in and out of the cell easily, and then others require special carriers, which are the proteins here. So we'll talk about that more specifically in a moment. I really like this picture a lot, this graphic, and let's dissect a little bit of what we're looking at. So let's start with our phospholipid bilayer here. If you notice, look at it closely. It's looking like this. Has this, right? Okay, we practice writing the lipid structures for this. This is why we learned how to do it. So what I want to do is take a minute and kind of just address the phospholipid bilayer part. So I'm going to click, and what I want you to do is to, okay, we left off like here, right, with showing those tails and the polar heads, right? They were like that. So in your mind, let's flip this way. Okay, so I'm just going to flip it in space. And then what I'm going to do is just grab some phospholipids that we practiced writing. I'm going to erase this. I'm going to grab some of the phospholipids that we have been practicing writing in these videos. So I won't go back through on why they're this structure. So for that, go back to our previous the previous video lectures. But so here's one phospholipid. Okay, these are the ones that have the fatty acids, right? So those are the tails. And then they have a polar head group. And then they're the phospholipids. Okay, so there's one structure that we've written. Okay, now I'm going to draw, and I think just to keep things kind of simple, I think I'll just draw the same one on the other side. And I really have to focus so I don't mess this up. Now, do you see how okay, and then now here's where I'm going to have a little bit of trouble because I don't have enough room. So I'm just going to do a more simplified structure on this side, if you don't mind. Just, you can say that there's another group on the end there. I just would have run out of space. Okay, so right now what we're looking at, and this is going to make some of our structure writing worthwhile, is here's the polar tails, right? Or it says non-polar tails. So what I want you to do every time you see this simplified structure, that you're actually thinking of all the structures you've learned how to write. And if you do, then that's really great because that means you're going to have a much better and deeper understanding of what we're talking about. So you see how those are the polar heads that I've circled in yellow and then the tails. And then what you should picture is, and it doesn't matter too much, but this could be the inside of the cell, and this could be the outside. That's kind of subjective on which one I pick. So this outside would have water, and the inside would have water. So you see how the polar head's going to be attracted to the water. And then what's holding together the inside here are nonpolar attractions. So those would be the London dispersion forces. Okay, now one more thing before we move on is that lipids are not, phospholipids and lipids, they don't want to form this structure. You put them into water and they're going to want to form that circular micelle structure. Go back in your notes that naturally they want to form circular structures where all the tails are in the middle and the polar parts on the outside. So this is not necessarily the natural structures that these guys want to form. And so helping this out is going to be our friend cholesterol, which we'll see in a few minutes in a later slide. Cholesterol is going to help to maintain these tails in a more rigid format. And then the other thing that we're going to see are proteins can be embedded in here. Now, I'm really excited about this part because it's going to have us rely on some things we've talked about in great depth, which is the structure and polarity of our amino acids. So touching the, so I didn't spell acid right, touching the part that is going to be near the hydrophobic tails on those lipids are going to be amino acids that are nonpolar, right? The nonpolar amino acids are going to attract the cholesterol. They're going to attract the hydrophobic tails. On the outside here, towards the water, those would be polar amino acids are going to stick out. Now, I know this is a blob, but I want you to think of that proteins are made up of amino acid sequences. And so, depending on what the protein's used for, we're going to talk more about how important that protein structure is to how it fits into this lipid bilayer. And also keep in mind that our proteins will have those distinctive features like alpha helices, beta pleated sheets. A lot of the proteins that are embedded in the cell membranes will have alpha helical structures, and we'll see that in a few minutes. So I hope that all the investment of time you've made in drawing these structures will help you to understand what is going on in a cell membrane. Because personally, I think just writing them like this up here is not very helpful for students. It's better to know what their structure is so you can understand the properties of the cell membrane. One other thing before we move on is if a substance has to get through this cell membrane, it has to go through the tails that are super hydrophobic. So one of the few things that can get through that easily will be nonpolar oxygen gas and CO2. So these are small nonpolar molecules that can get through the hydrophobic tail part of the cell membrane. Other molecules that are polar are going to require proteins to allow them to come through. And we'll come back to that in just a few moments. Okay, so I'm going to click backwards here. So we explored the phospholipid bilayer, and I'm going to erase this here. Okay, so we explored the phospholipid bilayer and how we should be thinking of it. Here's our protein that is shown as a blob, but we're going to see it as amino acids. When they're integral, they span the membrane. There's one called peripheral membrane proteins. That means that they're only on one side of the bilayer. The other things shown are the role of carbohydrates, sugars. Glucose, for example, can attach to lipids. Think back to the types of lipids lecture. There was one that showed glucose as being able to be attached to phosphate on a phospholipid. And so here, see how it's attached to the lipid bilayer that's in brown. Okay, so some glucose can go on lipids. One thing I did not stress in previous lectures is that glucose can also attach to proteins. So you have here, you see how carbohydrates are attached to that protein, and this. So these are long chains of sugars there. And those will serve important functions within the cell membrane, usually on the outside. So this would be the outside. And they tend to serve as flags that label as markers what kind of cell you're looking at, because there's distinctive differences between, let's say, neuronal cells versus hepatocytes, which are liver cells. So there's different kinds of cells, and so they'll have these different signature carbohydrates on the outside of them. Also important in cell-to-cell interaction. Okay, so a lot of what I just stated is going to be highlighted here. So membranes are sheet-like structures. You have the lipid bilayer here that we've just practiced drawing. It has what's called the hydrophobic core, which would be this part here. And those are going to be attractive forces in the hydrophobic core will be London dispersion forces. The thickness of the membranes is anywhere between 6 nanometers and 10 nanometers. An older unit is an angstrom, see that? So those are, it's just an older unit of measure. Membranes will have mostly lipid and proteins, which with some carbohydrate moieties. So when you have carbohydrate like glucose attached to a lipid, it's going to be a glycolipid. And a sugar attached to a proteins is a glycoprotein. And as we saw from the previous picture, it may not just be one sugar molecule, it might be a whole bunch. Okay. The next sentence is that lipids are a barrier to the flow of polar molecules. So things that cannot get in without help would be like glucose, as an example. We'll need special transporters for that. Molecules that can get in, so can easily diffuse, would be O2 and CO2. So these will diffuse because they are small and nonpolar. Okay. Proteins within the cell membrane. I mentioned one example there about transporting molecules through that, like glucose, but there's different functions. So what we're going to be exploring in a later video are pumps. So how are ions pumped in and out of the cell membrane? Protein channels that would allow for things like water to flow through, receptors, hormones that will trigger events within the cell are very important in metabolism. So the cell membrane will have receptors for things like insulin or so insulin's a good example. Energy transducers, these are going to be discussions of how signals are sent via in the brain, for example, through changes in energy potentials and voltages, and then enzymes. So we are going to look at examples of all of these in the next video lecture. Okay. Membranes are non-covalent assemblies, so that the proteins embedded in the lipid C are held together by non-covalent, so they're not anchored. So there's lateral movement within the cell membrane. And then membranes are asymmetric, meaning that one side of it, if you sliced it in half, they wouldn't look the same on both sides because remember they have a bunch of different molecules, macromolecules embedded. Fluidity is an important term. So lipid molecules diffuse rapidly in the plane of the membrane unless they are anchored by specific interactions. They don't rotate across the membrane. So membranes tend to be regarded as two-dimensional solutions of proteins and lipids. Okay, and there's also going to be some electrical polarization discussions, but we'll come back to that in our next PowerPoint. Okay, we're heading into the role of cholesterol in the cell membrane. I know you've heard of cholesterol with regards to heart disease. This discussion will put cholesterol in a much more positive light. So remember from our fatty acid discussion that saturated fats have only single bonds. Unsaturated fats have cis double bonds, and they can either be monounsaturated or polyunsaturated. So if we look over here with the unsaturated fatty acids that could be part of the phospholipid, they will not be as close together to one another because there's a kink in the chain due to the cis bonds. And what this does is it causes greater fluidity, less organization within the cell membrane. So what will happen is that cholesterol, so here's cholesterol, will embed itself within the cell membrane. And what it does is it forms some has London dispersion forces or van der Waals interaction between itself, because it's these big bulky rings, with the saturated fats within the cell membrane. And what that does is it keeps them more extended, more as a straight chain, and therefore keeps kind of the overall structure of the cell membrane. Temperature plays a role in the lipid bilayer. With heat, membranes become more disordered. And from an experimental viewpoint, there's a melting temperature, the membrane, where the membrane transitions from this very nice looking lipid bilayer to a more liquid crystalline structure. Okay, so the more the membrane has longer chain fatty acids and more saturation, more saturated fats, the higher the temp is needed to transition it from gel to fluid crystal. Okay, so membrane proteins, again, their functions could be a receptor, could be an enzyme, could be a transporter, and we will look at examples of all of those in the next PowerPoint. But for now, let's just take a look at just some different types of membrane proteins. So if we look at this, we have an integral protein number 1, okay, that's spanning and interacting with the hydrophobic parts. So 1 and 2 and 4 are all integral proteins. And then you have protein number 3, that's a peripheral, that's just on one side. And you can have peripheral proteins either on the inside of the cell or the outside. Some proteins can be anchored to the membrane, so this would be more of a covalent type interaction. What I like about this picture is that now we're moved away from the blob structure. So see this here. Those are the alpha helices of a integral protein that one has 1, 2, 3, 4, 5, 6, 7 alpha helical structures in there. Okay. And notice that on the cytoplasm side, so cytoplasmic side would be the inside of the cell, extracellular membrane would be on the outside, that one of the amino acid chains is going to link up to fatty acid chain on the lipid bilayer. And so those are some special types of anchoring that keep that integral protein in a certain spot. I don't normally stress this, so don't worry about all these fancy names here. But I just want you to realize that there can be some covalent modifications to hold the proteins in place. Here's another picture of different types of proteins. And it just kind of is trying to give you sort of that 3D picture. Let's take the OMPX one here. That's an E. coli. See this part here? Those would be alpha helices. And then you see here, those ones are the beta pleated sheets. And you see how that protein is spanning the entire cell membrane. Okay. And they just have, all of them have different looks to them. Some are a little bit bigger than others. In the case of down here, you see where it's a reductase. That's going to be an enzyme. So that's a very large, fancy integral protein. Okay. The cytochrome here, we're going to talk about cytochromes later. That's going to be a proton pump. It's going to pump charged ions from one side of the membrane to the other. Okay, so those are examples, and don't worry so much about the specifics on the slide. This one in particular was to kind of give you a feel for kind of moving beyond the blob and understanding that those are amino acid residues with those important secondary structures we learned in a previous chapter.